Hello everyone, welcome to our YouTube platform. In this video, we are going to discuss about the allosteric enzymes. In our previous video, we have discussed about the Michael-Menten enzymes, their regulation and types of inhibitors. So, if we compare these allosteric enzymes with the Michael-Menten enzyme, in case of Michael-Menten enzymes, these enzymes they obey the Michael-Menten equation, and these are the monomeric enzyme made up of one subunit. While the allosteric enzymes they obey the Hill equation, and these are oligomeric made up of more than one subunit. Now, in case of allosteric enzymes, there are two terms. One is the allosteric sites and other is known as the cooperativity. बात करते हैं एलोस्टेरिक साइट्स की. एज वी ऑलरेडी स्टेटेड कि जो एलोस्टेरिक एंजाइम्स होते हैं दे आर मेड अप ऑफ मोर देन वन सबयूनिट, so they have many other site beside the active site, and these sites are called as the regulatory sites or the allosteric sites, as we can observe in this figure. Similarly, these enzymes they shows the cooperativity. Now, what is cooperativity? It is simply the binding of substrate to one regulatory site. It will trigger the binding of substrate to all the substrate binding sites. This is known as the cooperativity simply. अगर बात करें ग्राफिकल रिप्रेजेंटेशन की, तो अपन ने देखा था माइकल मेंटेन एंजाइम्स के केसेस में कि वहां जो ग्राफ निकल के आ रहा था दैट वाज द रेक्टेंगुलर हाइपरबोला. Well, in case of the allosteric enzyme, we can observe a sigmoidal graph. Now, why it is a sigmoidal graph? बात करें इसकी, तो यहाँ पे एक लैग फेज देखने को मिलती है in this graph. Why this lag phase is there? Because जैसा कि अपने यहां पे बात की कि एक सबस्ट्रेट की बाइंड होने की देर है, इट विल ट्रिगर्स द बाइंडिंग ऑफ सबस्ट्रेट और टू द ऑल सबस्ट्रेट बाइंडिंग साइट. तो जब तक एक सबस्ट्रेट रेगुलेटरी साइट को रिकॉग्नाइज नहीं करता है, देन टिल देयर इज अ लैग फेज, और आफ्टर द बाइंडिंग ऑफ वन सबस्ट्रेट वी कैन ऑब्जर्व ए रैपिड इंक्रीज इन द एंजाइमेटिक एक्टिविटी. So that's why there is a lag phase. So this is the major difference between the Michael-Menten and the allosteric enzymes. Now the allosteric enzymes, these are the major regulatory enzymes of the biochemical pathway. क्यों major regulatory enzymes हैं? Because their activity can be regulated by the feedback inhibition. अगर प्रोडक्ट जो आप उस रिएक्शन से बनाने वाले हैं वो ऑलरेडी ज्यादा कंसंट्रेशन में आपके पास प्रेजेंट है, तो उस कंडीशन में उस रिएक्शन को नहीं होना चाहिए. दैट इज नोन एज द फीडबैक इनहिबिशन सिंपली. ये अपन एक एग्जांपल से भी समझ सकते हैं. देयर इज एन एंजाइम दैट इज कॉल्ड एज फॉस्फोफ्रुक्टोकाइनेज 1, आल्सो नोन एज PFK1. Now it is the first, or we can say it is the important enzyme of glycolysis, or we can say it is the first committed step in the glycolysis, where there is a conversion of fructose 6-phosphate into the fructose 1,6-phosphate on the expense of one ATP molecule. So there is a transfer, it is a transferase, there is a transfer of one phosphate from the ATP to the first position of this fructose, that lead to the formation of fructose 1,6-bisphosphate. But if the cell is already in the energy rich state, the purpose of glycolysis and the Krebs cycle is to generate the ATP in the form of, so generate the energy in the form of ATP. Now, if we already having the lot of ATP, then there is a feedback inhibition by this ATP of this phosphofructokinase 1 enzyme. So this is by this we can say that these are the regulatory type of enzymes whose activity can be regulated by the feedback inhibition. Now the cooperativity, it can be explained by two models. As we already stated in our previous slide that the allosteric enzyme, this shows the cooperativity. Now this cooperativity, it is of two types. One is known as the positive cooperativity, while the other is known as the negative cooperativity. Now, the binding of a activator to the regulatory site, if it changes the active site in such a way that it is able to recognize the substrate, then this is known as the positive type of cooperativity. While the binding of an effector molecule or the inhibitor to the enzyme, if it triggers the enzyme or changes the conformation of enzyme in such a way that it is now not able to recognize this substrate. Previously, it is able to recognize the substrate. Now, it is not able to recognize the substrate. It is an altered active site. Then it is known as the negative type of cooperativity. So, there is a positive and the negative cooperativity. Now, effector molecules, many effector molecules, they will bind to the allosteric enzymes and they will regulate the enzyme in the positive or the negative direction. Now, these effector molecules, they can be categorized into two groups. One is known as the heterotropic type of effector molecule, while the other is known as the homotropic type of effector molecule. If the substrate, it will itself act as an effector molecule, then it is known as the homotropic type of effector molecule. For example, if you can take the example of hemoglobin, जिसमें अपन दोनों ही explain कर पाएंगे homotropic और heterotropic effectors को. Now the oxygen is the substrate for this hemoglobin, and the binding of oxygen to one substrate binding site will trigger the binding of oxygen to all the substrate binding sites. Then the substrate itself acting as a effector molecules, that's why it is known as homotropic type of effector molecule. Well, if we talk about the heterotropic, there is again the example of hemoglobin. The carbon dioxide, it is not the substrate of hemoglobin, but still if there is a presence of carbon dioxide, it will not allow the binding of oxygen to the substrate binding site. So it is, we can say, if the effector molecule is something else than the substrate. Carbon dioxide is not the substrate for the hemoglobin, but still it is affecting the enzyme. So if the effector molecule is something else than the substrate, then it is known as heterotropic type of effector molecule. Next, we can discuss about some other type of enzyme, and first is the isoenzymes. Now, isoenzymes are the enzymes simply which catalyzes the similar reaction, जो एक ही तरीके की अभिक्रिया को संपादित करते हैं, अपन कह सकते हैं, बट दे आर एनकोडेड बाय द डिफरेंट जीन्स, पर अलग अलग जीन से वो बनते हैं. ऐसे एंजाइम्स जो अलग अलग जीन से बनते हैं पर एक ही तरीके की अभिक्रिया संपादित करते हैं, कैटलाइज करते हैं, देन दे आर नोन एज द isoenzymes. फॉर एग्जाम्पल, वी कैन टेक द एग्जाम्पल ऑफ लैक्टेट डिहाइड्रोजिनेज. करता क्या है ये एंजाइम? लैक्टेट को पायरुवेट में कन्वर्ट करता है. इस एंजाइम की अगर बात करें तो हार्ट मसल्स में ये एनकोड होता है, सपोज करें H4 जीन से. वहीं अगर बात करें मसल्स के अंदर इसकी, तो वहां पे ये एनकोड होता है आपके M4 जीन से. सो डिफरेंट जीन्स आर एनकोडिंग द सिमिलर एंजाइम्स which are encoding the similar reaction, we can say. So what is the use of these different enzymes which carries out the similar reaction? Now it is one of the way to regulate the physiological activity of the cells as per the requirement. It means these enzymes, they are either having the spatial separation, suppose one is functioning within the heart, other is functioning within the muscle, or they are having the temporal separation. एक कभी एक्सप्रेस हो रहा है, एक कभी अपना काम कर रहा है. तो या तो समय का इसके अंदर डिफरेंस रहेगा या स्थान का या प्लेस का डिफरेंस रहेगा. So it is, we can say, one of the way to regulate the physiological activities of the cell. Now other importance of this isoenzyme is that we can easily observe an internal injury. सपोज करें कि अगर ब्लड स्ट्रीम के अंदर H4 टाइप्स के आपको क्या एंजाइम्स देखने को मिल जाते हैं, इट मींस जो इंटरनल इंजरी है वो हार्ट में है और मसल्स डैमेज है और आपको M4 देखने को वेरिएंट देखने को मिल रहा है, दैट मींस जो डैमेज है वो मसल्स के अंदर है. सो इट इज वन ऑफ द वे टू आइडेंटिफाई द इंटरनल इंजरी ऑल्सो. नाउ नेक्स्ट इज द एलो एंजाइम्स. These are the enzymes that catalyzes the similar reaction encoded by the different genes. These are the enzymes which encoded again the similar reaction, but these are encoded by the different alleles of the same gene. एक ही जीन के दो अलग अलग अलील से एनकोड होते हैं, उनको अपन कहते हैं एलो एंजाइम. फॉर एग्जाम्पल द ग्लूकोज 6 फॉस्फेट dehydrogenase enzyme. Now, this enzyme it plays an important role in the HMP pathway, जिसके बारे में अपन आगे बात करेंगे. It plays an important role in the production of reducing energy, that is the NADPH2. Now, this is also required for the maintenance of we can say structure of RBCs. In the absence of this enzyme, that is the glucose 6 phosphate dehydrogenase, there is a more production of H2O2 that can lead to the hemolytic anemia. So, to prevent the hemolytic anemia, the presence of this glucose 6 phosphate dehydrogenase is necessary, and the genes for this enzyme is located on the X chromosome. Now, we are having more than 400 types of variants of this particular gene within the population. So, alloenzymes, these are simply we can say enzymes which are encoded by the different alleles of the same gene. Gene जो आपके located है कहाँ पे, X क्रोमोसोम पे, किसके लिए, ग्लूकोज 6 फॉस्फेट डिहाइड्रोजिनेज के लिए. पर एक पॉपुलेशन के अंदर अपने को इसके कितने वेरिएशंस देखने को मिलते हैं, मोर देन 400 टाइप ऑफ वेरिएशंस कैन बी ऑब्जर्व. Next is the Abzymes. These are simply the monoclonal antibodies, or we can say the antibodies having the catalytical activity. And last is the Ribozymes. As we already stated that the most of the enzymes, they are protein, or we can say all enzymes are protein except the ribozymes, and these ribozymes, these are the catalytical RNA. It was first discovered by Thomas Catch and Sidney Altman, firstly discovered in case of the tetrahymena, where they are involved in the process of rRNA splicing. The best example is the peptidal transferase, that is the 23S rRNA, which transfers the peptidal chain टू द एक्सेप्टर डोमेन, जिसके बारे में अपन आगे बात करेंगे जब अपन बात करेंगे molecular biology. So 23S rRNA in the ribosome, that is also known as the peptidal transfer. It is the best example for ribozyme. So this is all about the allosteric enzyme as well the isoenzymes, alloenzymes, Abzymes and ribozymes. Thank you very much.